Today's case study is cross-dressing or crossing over, sex testing in women athletes. So let's start off with the situation. Santi's story. So Santi won the silver medal in the 800-meter race at the 2006 Asian Games in Qatar. This, by the way, is a true story, so we're not making this stuff up. So following her silver medal win performance, her sex was officially questioned. So there was an official question of whether Santi was actually female, and if she was not female, she should not have been entered in the event and been able to win. So here are a couple announcements, indications, and stuff from December 18th, 2006. Top Indian woman athlete, Santi, who won a silver medal at a recent regional championship, has failed the gender test, according to official reports. Sports writer K.P. Mohan said that a team of doctors, including a gynecologist, endocrinologist, and psychologist, normally examine athletes and put them through physical and clinical examinations during a gender test. Santi's test was done soon after she came in second in the women's 800-meter race on December 9th, but it's not clear how she failed the test at the Asian Games in Doha. So in this particular case, what we have is we have a real-world event where a woman who won the silver medal was accused of not actually being female. Tests were run and Santi was disqualified. Okay, the gender test revealed an anomaly. The details of exactly what the test revealed and everything were not given. However, this case study is based on the idea of what could have gone wrong or what could be involved. Was one side correct, the other side wrong? Was something lying? Or is it possibly a little more convoluted and nuanced than that? So let's take a look and learn a little bit about the biology behind this. So how do you know if someone is male or female? So what I'd like you to do is several points throughout this presentation. I'm going to ask you to pause the video and try to answer a question or a series of questions. So this is our first one. What I'd like you to do in just a moment, I want you to pause the video and try to identify at least 5 characteristics of any type that you might use to distinguish between males and females. So what makes males and females different? It can be anything that you can think of. So go ahead and pause the video. As soon as you're done pausing, come on back, start the video again, and we'll continue talking about this. So go ahead and pause now. Okay, you're back. Hopefully you've come up with at least 5 characteristics. So we're going to start by talking about how sex is determined in humans. There are a couple different ways. The first one, probably the most common, and probably includes some, if not all, of the traits you identified, is primary and secondary sexual characteristics, looking at the anatomy, differences between males and females. Okay, genitalia. Okay, so we have different genitals in males and females. We have different body hair. For example, men have facial hair, women generally do not. The pelvis is different. You can have researchers who study the anatomy of humans can actually look at the bones, the pelvis, or the hips, and can tell if the skeleton is male or female based on measurements there. And there might be other things you came up with. Another way we can determine sex is by looking at the physiology. So kind of look at the chemistry happening inside the body. So the function and interaction of the sex organs, including concentrations of different sex hormones. Like females tend to have higher levels of estrogen. Males, on the other hand, have higher levels of testosterone. And there might be other physiological things going on that separate males and females that you might have listed. So here's our first question for you. Imagine that you are a member of the committee assigned to determine where Santi is female. Here are some results from the initial tests. Understand these are not Santi's actual results. We don't know those, but these are representative of what might have occurred. So these are the results you see. Female genitalia, yes. Breasts and pubic hair, yes. A regular menstrual cycle, never. It says never had them. Based on this information, would you conclude that Santi is male or female? Go ahead and pause the video, make your decision, and then restart. Okay, so what did you come up with? Odds are pretty good you went with female in this case. You might have gone with male, but you probably went with female because when we look at this, female genitalia, breasts, and pubic hair, those two strongly suggest female. However, no regular menstrual cycle. that might be leaning to you, maybe something isn't female, except for one possible problem. High performance athletes who spend so much time training, female athletes, often do not have a regular menstrual cycle or don't have any menstrual cycle at all. The amount of physical activity they're involved in often disrupts the menstrual cycle. So that in and of itself is not an indicator that something is not female. So in this case, the signs indicate female is most likely the correct answer. So is there another way that sex is characterized in humans? And there is. And that's by looking at chromosomes. Okay. Chromosomes are the DNA found in cells. And female humans possess 2 X chromosomes in each of their cells. So they have 2 X chromosomes. Males, on the other, have 1 X chromosome and 1 Y chromosome inside their cells. So by looking at the chromosomes, we have another way to determine if an individual is male or female. Which chromosomes do they have? Well, how can we actually visualize chromosomes, and how can we look at them? Chromosomes, which we will talk about in much more detail later on, but for right now, chromosomes are only visible when the cell's nucleus prepares for division. And so what we do, we have this process where we wait for cell division to happen, and then we can stain the chromosomes, photograph them, and take a look at what they look like. Before we actually see a picture of what chromosomes can look like, let's go ahead and talk about chromosomes in a little more detail. So during nuclear division, DNA is tightly packed. Okay, so the DNA, it wraps up and it gets what we call condensed. It becomes highly visible. And you can see a picture of that here. So this is our nucleus, and we have a bunch of DNA inside of it. And if we zoom in on one of those DNA, this is what it looks like. Okay, the DNA, and you can see it drawn here. This is kind of an X. Okay, now this is something really, really important, and it might be a little confusing, but we'll make sure to clarify in this lecture as well as some others. But chromosomes actually are 1 strand of DNA, not 2 like you see here. However, right before cell division happens, the chromosome undergoes what's called DNA replication. It makes a perfect copy of itself, and when it does that, the 2 copies, perfect copies of each other, are connected to each other. So this makes a chromosome. So this is a chromosome, and this is a chromosome. Just one's copied to the other. So when you see chromosomes drawn as X's, it simply means DNA replication has occurred. And one more important thing, the naming. This, as I said, is a chromosome right here. This is also called a chromosome. When replication and you have the X, the chromosomes are made of 2, what we call sister chromatids. And you can see that name right here, chromatids. So the chromosome is composed of 2 sister chromatids, okay, which are all made of DNA. So let's continue here. This is what we call a karyotype. A karyotype is when a geneticist stains and photographs all the chromosomes inside a single cell. So this picture are all 46 chromosomes that occur in one human cell. When you look at them, certain patterns should start showing up when you pay attention. The first thing, you can see they're paired off and they're numbered. So you have numbers 1 through 23. Okay, so we have 23 pairs of chromosomes. We have 2 chromosome number 1s, 2 chromosomes number 2, 3, 4, and so on. If you look at the banding pattern of the 2 chromosome number 1s, you'll see they're the same. There's a green glow at the upper edge, a red dot in the middle, kind of a mix of green and reds at the lower part. If I look at number 16 here, there's green on both ends and red in the middle, and so on. The reason there are pairs of chromosomes in each of your cells is because the pairs come from each of your parents. You got one chromosome number 1 from your mother and the other chromosome number 1 from your father. The same thing with each of the other chromosomes. So you effectively got 23 chromosomes, one of each number from mom, and 23 chromosomes, one of each number from dad. We call these pairs homologous chromosomes. Okay, homologous chromosomes. And that term is going to be important. You're going to hear that over and over, so remember it. The other thing, though, that I want you to really note here is the last pair of chromosomes down here. These are what are called the sex chromosomes. And when you look at them, if they are both the same, that means they're both X chromosomes. On the other hand, if they are different sizes, one of them's large and the other one is small, that is an X and a Y chromosome. In this case, we have two X chromosomes. They're both large, which means this individual is female. So now let's test your understanding of this and another question. So here's another karyotype. This karyotype was performed on an athlete. Here are the results. Based on this karyotype, is this individual, this athlete, male or female? Go ahead and pause for a sec, think about it, and then get your answer and start the video again. Okay, hopefully you said that this is a male, A. And if you did, you are correct. The only chromosomes that matter when determining sex are the last set right here. And you can see there's one X and one Y chromosome. And one of them is long, that's the X, and one of them is very small, that's the Y. If it was XX, they would both be the same size. So you'd have two chromosomes the same size. So this would be a male. So we now have several different ways to identify if someone is male or female. We can look at their anatomy, we can look at their physiology, or we can go in and actually look at their chromosomes to determine if they are male or female. So, another question. If Santhi is normal female, what would her karyotype be? Go ahead and pause, come up with your answer, and then play. Okay, hopefully, if Santhi's normal female, hopefully you said A. She would be XX. That would be female. XY would be male. YY would not exist. They would not be born. And XXY actually does exist. However, biologists generally view it as if a Y chromosome is present, that means they are male, even though there are two X chromosomes here. Okay, so this does occur on occasion, but a normal female karyotype would show A, XX. Okay, sex. Have your attention now? Good. Let's go ahead and get into reproduction. So, genetic material during reproduction. You contain genetic material both from your mom and your dad. And as I said before, you have 46 chromosomes inside yourself, 46 strands of DNA. And so will your children. Your children will also have 46 chromosomes. How does this happen? Well, in very basic terms, males and females both produce what we call gametes or sex cells. In the case of males, those are sperm cells. For females, those are egg cells. We are then going to merge an egg and a sperm together in a process called fertilization to produce a zygote. A zygote is an egg and a sperm joined together, so DNA from mom and dad, and that zygote will then begin to divide over and over and become a child. Okay? And that child will then grow into an adult and can have children of its own. This is the process of what we call sexual reproduction, where we bring genetic material from two individuals, merge them together to produce an offspring. Earlier in class, we talked about one form of cell division, mitosis. And mitosis can be used for reproduction, but it has a serious problem when we start trying to talk about sexual reproduction. So let's look at the problem and understand why we can't use it. So here, I am going to have, here's mom and here's dad. Okay, These are just cells with mom and dad. And I've already told you mom and dad both have 46 chromosomes. Okay, great. Well, in mitosis, mitosis goes in and it produces perfect copies of this, 2 of them. So we're going to go in, we're going to produce egg and sperm cells. So this will be dad. So we're producing a sperm cell with 46 chromosomes and another sperm cell with 46 chromosomes through mitosis. Mom is going to produce 2 egg cells, same process. These will each have 46 and 46. And now sexual reproduction happens where we are going to take this sperm cell and this egg and merge them together. Fertilization, and we're going to end up with an offspring that has 92 chromosomes. Then this individual is going to have children with another individual that went through this same process, 92. And these 2 individuals are now going to have an offspring with 184 chromosomes. This is not what happens in the real world. I have 46 chromosomes. My wife has 46 chromosomes. My children each also have 46 chromosomes. They don't have 92. My grandkids will have 46 chromosomes, not 184. If we use mitosis for sexual reproduction, every generation we would double the amount of DNA, the number of chromosomes in each cell. And eventually the cell just couldn't contain those chromosomes anymore. That is the problem of mitosis. And so evolution has found a solution. When we create gametes, sex cells, eggs and sperm, we do not use mitosis to make them. Instead, we use a different process called meiosis. Okay, so very general terms about meiosis here. We are going to start off, we have mom and dad. They each have 46 chromosomes. Dad is going to produce sperm cells. Those sperm cells are each only going to have 23 chromosomes, half of the full number. Mom is going to produce egg cells that also have 23 chromosomes, half of the original number. So mom's egg has 23, dad's sperm 23. They're going to merge together and form a zygote, which once again has 46. 23 plus 23 is 46. So the baby has the same number of chromosomes as mom and dad. This is accomplished through a process called meiosis. Okay, meiosis is another form of cell division. But I want to clarify 2 important terms that you're going to hear when we talk about this. Haploid and diploid. A diploid cell is a cell that has the complete set, 2 copies of the complete set of chromosomes. So remember when we looked at that karyotype, let's go back and look at it now. When we looked at that karyotype, there are 23 pairs of chromosomes. So 1 complete set of chromosomes is 23 chromosomes, 1 of each one. That would be 1 complete set. When we have a diploid cell, that cell has 2 complete sets of chromosomes, or for humans, 46 chromosomes. Haploid cells, on the other hand, only have 1 complete set of chromosomes, 23 chromosomes. So it has 1 number 1, 1 number 2, 1 number 3, 1 number 4, and so on. Diploid have 2 number 2s, 2 number 3s, 2 number 4s. Okay. So these terms, haploid and diploid, are about to become very, very important as we go through this process. Now, let's clarify something about chromosomes. Okay. A chromosome can be either a single strand of DNA or 2 strands of DNA from DNA replication that are still attached together. These are each 1 chromosome, 1 chromosome. Okay. Now, a haploid cell will have 1 of each chromosome in it. That chromosome might look like a single strand, or it might look like a double strand with 2 chromatids, but they're connected, so we count it as 1 chromosome. So for each of the different chromosomes, we only have 1 copy. A diploid cell, on the other hand, has 2 copies. So this is chromosome number 1. And you'll note I have a blue version and a red version. One came from mom, one came from dad. When DNA replication occurs, they both are sister chromatids. They're paired, but I still have 2 copies of it. Haploid, only 1 copy of it. Okay, so haploid, 1 complete set of chromosomes. Diploid, 2 complete sets of chromosomes. By taking cells and going from diploid to haploid, we avoid doubling the amount of chromosomes every generation. So let's answer another question and let's see how this works. Okay, so assume that this is one of Santi's chromosomes in the picture here. This chromosome is composed of 2 chromatids joined by a centromere. That's the point where they're joined together. So the centromere is here. So this is basically the X I was drawing before. Based on that, which of these is correct? Go ahead and pause, read through them, and come up with your answer. Okay, so which one did you come up with? Let's run through each of them and see what sounds good. So A, these chromatids make up a diploid chromosome. The term haploid and diploid refer to the set of chromosomes, not a single chromosome. So that answer does not work. You can't have a diploid chromosome. You can have a diploid set of chromosomes. This B, the cell that contains these sister chromatids must be diploid. Well, a minute ago we looked and said haploid cells can be either single or replicated copies. So this could be haploid or diploid. We do not know. B might be right, but when I said it must be, that's the problem. Okay. C, the sister chromatids were formed by replicating a single chromatid. That is correct. We had 1 strand of DNA and we made a perfect copy of it, replication, and now we have 2. And then finally D, the sister chromatids were joined by fertilization, bringing together a maternal and paternal chromatid. Nope, nope, nope. Fertilization is when we bring an egg and a sperm together, not when we replicate DNA. So D is wrong. So the correct answer is C. Let's carry on. Meiosis. Okay, the purpose of meiosis in animals. Animals produce gametes with just half of the parent's genetic material. So gametes, another word for gametes is sex cells. In animals, we call these eggs and sperm. In other organisms, they might be called different things. For example, plants, sperm cells are pollen. Okay, so they might have slightly different names. But gametes are always haploid. They have half the total number of chromosomes that a diploid cell has. So basically, how does meiosis work? Well, meiosis creates gametes that with only 1 of each pair of homologous chromosomes gets into each gamete. So remember before I said that we have 2 chromosome number 1s, 2 chromosome number 2s, 2 chromosome number 3s. Well, in a gamete, it's going to get 1 of the chromosome number 1s, 1 of the number 2s, and 1 of the number 3s. Okay? And in the end, the gametes from 2 parents are going to fuse together, restoring the original number of chromosomes because the egg has half of them, the sperm has half. They come together, add it up, we've got the full number in the zygote. So how does meiosis work? Well, here's kind of a cool thing. You've already learned about mitosis. Meiosis is basically mitosis twice, with a couple little additions thrown in. So meiosis produces 4 haploid cells. So we're going to get 4 gametes out of this from 1 diploid cell. And we're going to go through 2 processes, 2 rounds of cell division. The first one is called Meiosis I. The second is Meiosis II. At the end of Meiosis I, we will have 2 cells, and those cells will be haploid. We will then do a second round of cell division, Meiosis II, that are going to split the 2 sister chromatids into a single chromatid in each cell. Let's take a look at how this works. This is Meiosis I. And if you look carefully, you'll see it's very similar to mitosis. We have some of the exact same stages that you learned. We have interphase, we have prophase, we have metaphase, we have anaphase, we have telophase. They're the exact same steps. And for the most part, the same things happen. So you actually know most of this stuff already. Okay, but let's go into a little more detail and add some details. So meiosis I, meiosis only occurs in sex cells. Cells that are destined to become gametes are the only kind of cells that undergo meiosis. The cells in my skin do not undergo meiosis ever. Okay, but so cells from a sex organ, the ovaries or testes in animals, they're in interphase. It comes time to divide. What we are going to do, all our chromosomes are going to gather in there. They are going to condense, just like in mitosis. You can see them represented as X's. However, what I want you to pay attention to here is they have drawn different colors, but also different sizes. We have two large X's. One of them is black, one of them is gray, and two smaller X's. One of them is black, one is gray. Okay? The black set, large one, small one, represent chromosomes from dad. The gray ones, one large, one small, represent the chromosomes from mom. Now understand, your cells have 46 chromosomes in there. These cells only have 4, but it's okay. We're just simplifying it. You could do this with 46, but it'd be very hard to draw and see what's going on, so we've simplified the cell. Now, something I want to do right now before we start, how many chromosomes are inside this cell right here? Count the X's. We have 4 chromosomes. Remember that. We're going to come back to it. Okay, now, what's going to happen here is the homologous chromosomes are going to find each other. So our homologous chromosomes, the 2 big X's, one gray, one black, are going to find each other. The 2 small X's are going to go find each other, and any other pairs will find each other. Okay, the homologous chromosomes pair up. Then, that's prophase. Metaphase, the chromosomes line up in the middle. But if you look carefully, the homologous chromosomes are together. Anaphase. Anaphase happens. Remember, mitosis, anaphase, the sister chromatids separate and start going apart. Okay, little different here though. The sister chromatids do not separate. This is a change. Instead, the homologous chromosomes separate. So one of the homologous chromosomes goes left, the other homologous chromosome goes right. Every pair of homologous chromosomes split, one going to the left, one going to the right. At the end of meiosis I, new nucleus forms around each of them, and cytokinesis, the cell divides. I want you to notice a couple things. First off, how many cells do I have at the end? I have 2 cells. How many chromosomes are in each of those cells? How many X's? 2. So we just went from 4 chromosomes to 2 in each cell. We just went from a cell that is diploid to a cells that are haploid. Okay? The other thing I want you to notice, because this is really important, each new cell contains 1 large X and 1 small x. 1 large X, 1 small x. That's meiosis I. The purpose of meiosis I is to take all the homologous pairs and separate them. So 1 homologous chromosome goes that way, 1 goes that way. That's its purpose. As soon as we finish meiosis I, we jump into meiosis II. We are basically going to do cell division a second time. But one really important thing, no DNA replication happens at this point. We do not replicate the DNA again. These are our 2 cells from the end of meiosis I. They're both going to do the same thing. They are going to go into prophase, condense, nucleus breaks down. Metaphase, they are going to line up along the equatorial plane. Anaphase, the sister chromatids are going to break apart and go to the different poles, just like in mitosis. Telophase, a new nuclear envelope is going to form and cytokinesis. What we end up with is 4 cells. How many chromosomes are in each of them? Well, we started off with 2 chromosomes over here. How many chromosomes do we have in each of these cells? Even though they're not X's anymore, they're still chromosomes, so we still have 2. So what we have done at the end is we have created 4 haploid cells. And that is the process of meiosis. Round 1 is all about separating homologous chromosomes. Meiosis II, the second round, is about separating sister chromatids to go from 1 diploid cell to 4 haploid cells. These cells will then go through a process to become either egg or sperm cells, depending if they're male or female. Okay, so another question for you. Which is true at the end of Meiosis I? Go ahead, pause, read through, come up with your answer. Okay, what did you guess? Well, the correct answer is B. The cells are haploid. That is correct. Let's look at the other ones and see what's wrong with them. So A, 4 cells have been produced. Okay, at the end of Meiosis I, that is not true. We only have 2 cells. C, the DNA will be replicated one more time. Nope, DNA does not replicate again. D, the cells are ready to perform as gametes. We're only halfway there. We have to go through Meiosis II for that to be true. And finally, each chromosome consists of a single strand of DNA. No, we still have the sister chromatids joined together, the X's. So the cells are haploid. During Meiosis II, which of the following is true? Go ahead, pause, read them, and then come back. Okay, in this case, let's just run through them. Homologous chromosomes separate. Nope, that happened during Meiosis I, so not A. B, the DNA is replicated. Nope, that happened in Meiosis I also. C, the gametes fuse together. That happens in fertilization, not during meiosis. D, the sister chromatids separate. Absolutely correct. Yes, D is the correct answer. And since we ruled out A, B, and C, E cannot be true either. So D is our correct answer. Okay. Why on earth do we do this? Sexual reproduction. Oh my goodness, this sounds really confusing and problematic. Mitosis was so much easier. Why do we do this? Because sexual reproduction in the process of meiosis can produce genetic variation. And genetic variation is absolutely essential for long-term survival of species living in environments that are constantly changing. Okay? So how does it create genetic variation? It can create genetic variation in a number of ways. Meiosis has 2 ways in particular we're going to focus on. The first one is what we call independent assortment. Every gamete that is formed is unique. They are not the same every single time. That is why if you look at any siblings you have, brothers or sisters, they look similar to you maybe, maybe not, but they don't look the exact same. So independent assortment is the reason for that. So remember I said that you have homologous chromosomes. You have one from mom, one from dad in each of your cells for each of your different chromosomes. And that's true for you as well as, or for your parents, mom and dad. Okay, so if I look at this, well, we just said during meiosis the homologous chromosomes are going to split. They're going to go one to the left, one to the right. Independent assortment means when I've got these 2 homologous chromosomes that you see here, an orange one and a blue one, one of them, as I said, is going to go to the left. One of them is going to go to the right. But which one? It is completely random. There's a 50% chance that this particular gamete is going to get an orange chromosome or the blue chromosome. 50% chance of each one. But to add even more variation here, the next set of homologous chromosomes, these ones, there's also a 50% chance that it will get the orange one or the blue one. Let's pretend for the first chromosome it got the orange one. Great. What are the chances now of it getting the orange one or the blue one for the second set of chromosomes? It's still 50-50. What that means, when we have 23 different chromosomes, we might get, for chromosome number 1, the orange one. For chromosome number 2, the blue one. Number 3, the blue one. Number 4, the orange one. And so on. You can see how with 23 different pairs, you can get a lot of different combinations depending which one you inherited. So some of your genes you inherited from your mom, and some of them you got from your dad or from your grandparents even. And that's true for every single gamete. Every time homologous chromosomes lined up, you have a 50-50 chance of getting either one. Okay. So here's a question. How many possible combinations of maternal chromosomes are possible in a human ovum due to independent assortment during meiosis? So how many possible combinations are there as far as what you could have gotten? Okay, so go ahead and pause, see if you can figure it out, then come back. Okay, so I want to remind you, you have 23 pairs of chromosomes. And for each of those pairs, okay, you can get either chromosome A or chromosome B. You have two options. The way to figure something like that out is you take how many options you have, so in this case 2, and you put it to the n power, okay, the number of different times you're going to do this. So this is the same as saying 2 to the 23rd, which is about 8 million different combinations. So the fact that independent assortment is shuffling around your genes and such, by independent assortment with 23 different pairs of chromosomes, you can produce approximately 8 million different unique egg or sperm cells. That is why you don't look the same as your siblings. The combinations are tremendous and a lot of variation occurs. So independent assortment is a huge way to get variation. Okay, so we talked about this a little bit already, but in mammals, males have one X and one Y chromosome. So males will be XY. Females will be XX. By the way, I specify here in mammals because other types of animals might be different. For example, birds, XY are females and XX are males. But don't worry, if you ever hear an example of this, it's going to just be humans or mammals, and so you can go with XX and XY, male, female. Okay, in males, half of the daughter cells formed by meiosis I are going to get the X chromosome, and half of them are going to get the Y, because this dad, okay, has an X and a Y. He's got a 50% chance of handing over the X and a 50% chance of handing over the Y. So half of the sperm cells the dad creates are going to have an X chromosome in it. The other half of the sperm cells the dad creates are going to have a Y chromosome in it. And that is going to influence what the offspring, what the child is going to inherit. It's going to inherit either an X or a Y chromosome. So how is sex determined in humans? Well, dad is going to contribute the sperm, mom is going to contribute the egg. We know dad, because he's male, has X and Y. We know mom, because she's female, has X and X. So that means dad has a 50% chance of delivering an X or 50% chance of delivering a Y. We know mom is going to deliver either this X or this X, but she's giving an X no matter what. This box you see here is what we call a Punnett square. We'll talk about these in more detail when we get into genetics, but it's a way to predict possible offspring. So if an offspring inherits this sperm cell and this egg cell, they will be XX. This sperm cell and this egg cell will be XY, and XX and XY. Now, if you look carefully, you will realize that these two boxes are going to produce girls, and these two boxes are going to produce boys. So there is a 50-50 chance that any offspring will be a girl or a boy. Okay? It's a random chance. Which parent determines the sex or gender of a child? The mom or the dad? Hopefully you said the dad, because the dad is the one that determines if a Y chromosome will show up or not. If he passes a Y chromosome. Mom can only pass an X chromosome on, so she doesn't actually determine the sex of the offspring. The dad does. Okay, another question. Remember Santi? We haven't talked about her for a while. We've been talking about meiosis. Let's jump back into this. So, could Santi have an XX karyotype and be male? So go ahead and pause, read through the choices, and come up with your answer. Okay, so what do you go with? Well, let's look at the answers here. So, no, an XX individual is always female. Based on what we've talked about, that could have been your answer. And I understand completely why you would have put that. B, yes, it's common. Well, no, it's not common. XX is female, not male. So B is not true. C obviously has some more information needed. So it's the correct answer. C is actually the correct answer. But you don't actually have the information to have known that. You might have said, he's trying to trick me, I'm going with C. But yes, if male determining controlled gene is carried on one of her X chromosomes. So the question that comes up here is, why does having XX chromosomes make someone female? Why does having an XY set of chromosomes make someone male? That's the question that becomes important. And so that's what we're going to get into now. Okay, before we do that though, we need to understand one other way meiosis can cause genetic variation. This is doing something that we call crossing over. If you remember, I told you that before the first round of meiosis completes, the homologous chromosomes kind of seek each other out, find each other, and group together. Okay? And we have drawn homologous chromosomes here, one blue, one yellow. The homologous chromosomes pair up, okay? And when they do that, it is not uncommon for them to cross over each other. By the way, when they pair up, we call that a tetrad. That means it's a pair of homologous chromosomes that have paired up together during the early parts of meiosis I. Okay, when they pair up, it is not uncommon for them to kind of overlap each other in areas. That overlap is called a chiasmata. Sometimes when a chiasmata happens, recombination occurs. So recombination is when the two pieces of DNA exchange. Basically, the blue piece from, I'm going to call these A and B. So the blue piece from B and the yellow piece from A switch places. And you can see that down here. Now, this is not a big deal in crossing over because homologous chromosomes contain the same genetic information on them, but they might have different versions of that information. So what I mean by that is, even though both of these chromosomes in, let's just say, right at this spot where I'm drawing, contain the gene for eye color. Okay, so what color eyes you're going to have is determined by a DNA code located in that position. But on the yellow one, it codes for blue eyes, and on the blue one, it codes for brown eyes. So when we recombinate, it's not a problem because we still have the eye color genes there. But now, chromosome A this time codes for brown eyes and chromosome B codes for blue. So they've kind of switched. And if we start doing that in different places, we can get even more unique combinations than independent assortment gave us. Because we're kind of breaking the chromosome, switching pieces up and putting it back together. Okay, so this is called crossing over, and it occurs all the time in meiosis. Okay, so by crossing over, we increase the variation dramatically, even beyond what independent assortment gives us. Okay, so this is going to become important in just a minute. Okay, so crossing over can occur anywhere along autosomes. Autosomes is a term for all the chromosomes that are not sex chromosomes. So if you remember back to that karyotype, chromosomes 1 through 22, those are the autosomes. The sex chromosomes, just the X and Y chromosomes, they can cross over also. However, they only tend to cross over at the tips. And the reason for that is the X chromosome is gigantic and the Y chromosome is tiny. So they usually only cross over on the very extreme edges. They don't cross over very much. The X chromosome contains a large number of different genes, but the Y chromosome contains very, very few genes. It's tiny. It doesn't have very many. And that's going to come into play again in just a minute. So, during crossing over, all of the following happen except, which one's wrong? Go ahead and pause, read through, and come up with your answer. Okay, let's run through them. The homologous chromosomes line up in pairs. Well, that's true. They do. Okay, that's when they form that tetrad and such. B, the homologous chromosomes swap pieces with their adjacent partner. Well, yes, that's what crossing over is. So that's true. Remember, we're looking for what's not true. C, crossing over decreases the genetic variability in gametes. No. It increases variation. The gametes are going to become even more different from each other because of crossing over. So that's the correct answer. That is the exception. And then finally, D, each chromatid is unique after it is crossed over. Nope. Some chromatids are unique, but some of them don't remain unchanged. If we go back and look at that previous image, there we go. While this chromatid and this one did change, these other two on the outside did not. Okay, so cross out A, B, and D. C is our correct answer. So how is sex characterized in humans? We have talked about anatomy, physiology, and chromosomes, but there is a fourth answer, genes. Okay, we will talk about genes in more detail coming up very soon in class. However, a gene is a section of DNA that codes for a protein, and it produces an effect inside your body. Okay, the reason you develop as male or female is because certain genes in your body are present and are turned on or off at times during development, and that causes you to develop as male or female. Okay, so genes influence if you are male or female. And we talked earlier about X chromosome, Y chromosomes. There are different genes located on the X chromosome and the Y chromosome. So as a result, these genes are going to influence what you develop into. One gene in particular is the one we're interested in. It's what's called the SRY gene. And here it is. The SRY gene is located on the Y chromosome. Okay? It's one of the few genes located there. And in early development, everyone starts developing as females. Okay, there's no distinction between male and female humans for the first 7 weeks of development. Okay? However, something changes at the 7th week. Males who contain the Y chromosome and the SRY gene is activated in the 7th week of development. And what the SRY gene does, it in turn activates other genes. And those genes then help develop gonads and testes and other characteristics that make an embryo develop into male. Females don't have an SRY gene, so no SRY gene. So their gonads develop into ovaries by default because they're not getting those other genes turned on, even if they're present. So the SRY gene is present on the Y chromosome. It activates other genes and turns on male characteristic genes. Females don't have that gene, and so females, no Y chromosome, develop as female. So that is why having different chromosomes causes someone to be male or female. So the question is, does this process sometimes change or mess up? So here's what we're going to do. I have a video. I'm going to try to play the video, and hopefully sound records and everything. So give me a second, and we're going to go ahead and play this and see what happens. It's going to talk about the SRY gene and when something can happen.
Under normal circumstances, a human embryo contains 2 sex chromosomes, one from the mother and one from the father. If the embryo receives an X chromosome from its mother and an X chromosome from its father, the baby will emerge as female. If the embryo receives an X chromosome from its mother and a Y chromosome from its father, the baby will emerge as male. By popular belief, it's not the Y chromosome itself that determines the gender of an embryo. It's only a small segment of the chromosome, called the sex-determining region Y, or SRY gene, that determines whether or not an embryo will develop testes. Remember how I said at the beginning of the video that under normal circumstances an embryo with 2 X chromosomes is female and an embryo with an X chromosome and a Y chromosome is male? Well, that's not the truth in all cases. put it plainly, if the SRY gene is present in an embryo, it will stimulate the development of the male reproductive system. If the SRY gene is absent, the embryo will instead develop as female. It just so happens that this gene can be misplaced onto an X chromosome, resulting in a male embryo with two X chromosomes. This is called XX male syndrome, and individuals with this condition are phenotypically male. The term phenotype refers to the observable traits of an organism. The same thing can happen in reverse. With Swyer syndrome, an individual has one X chromosome and one Y chromosome, but the subject is phenotypically female because the Y chromosome lacks an SRY gene, or the gene is present but non-functioning. Note that there are other mutations or genetic disorders that can cause chromosomal abnormalities. The term aneuploidy, for example, refers to a type of chromosomal abnormality in which a cell is either missing or has extra copies of a chromosome. In the case of trisomy, a female can have 3 X chromosomes and a male 2 X chromosomes and 1 Y chromosome, or 2 Y chromosomes and 1 X chromosome. Even rarer is tetrasomy, the presence of 4 sex chromosomes as opposed to the regular 2. In females, there's even a syndrome called pentasomy X, in which the individual has 5 X chromosomes. As expected, individuals affected with this condition typically suffer from serious health defects.
So in the video, it identified sometimes the SRY gene moves. How can it move? Crossing over. So if crossing over occurs between an X and a Y chromosome, one of several things can happen. One, no effect at all, okay, in which case we just have still XX or XY determines male or female. But it is possible for the SRY gene to move from a Y chromosome through crossing over onto an X chromosome, in which case the individual would be XX, but we would say they are SRY positive because they have the SRY gene on one of their X chromosomes. If that happens, would that individual appear to be male or female? They would appear to be male. They would develop because they have the gene that activates all the male characteristics. Or you might have someone who's XY, but because of crossing over, their Y chromosome lost the SRY gene. That person would be SRY negative. So even though they have a Y chromosome, they don't have the SRY gene. Would that person be male or female? That person would develop as female. So what you can see here is the presence of an X or Y chromosome isn't 100%. 1 out of 20,000 births experience crossing over that affects the location of the SRY gene, and that can influence the actual sex of the offspring. So CQ10, here we go. If you were a member of the Asian Games Medal Committee and Santi's karyotype revealed that she is XY and SRY minus, what would you do? Go ahead, read through the answers and make your choice, and then come on back. Okay, so here we go. Answer A, if she had female genitalia, you'd allow her to keep her medal. B, she's genetically male, take her medal away. Or C, perform additional tests. There is no one right answer here. You've been taught the biology now of how someone can be XY, and even though they have a Y chromosome, it is possible that they've developed as female because they do not have the SRY gene. That might be what's happening here. Okay? This is the type of question that governing boards and stuff have to try to deal with, and often understand you have more biology knowledge than they do, as they're trying to understand and make acceptable decisions and stuff for this. Is important and often challenging for them. Okay? Next question. What do you think about requiring gender testing for female athletes in international competition? Tell you right now, there's no right answer here. What do you think is appropriate? Go ahead and pause, read through, and make your choice. Okay, let's take a look. In A, gender testing. It should be banned because gender determination is so complex. We've seen some ways that it's much more complicated than you think. So banning gender testing all around could be one option. B, it's necessary to ensure an even playing field. C, it is necessary but needs to include a large number of genetic tests to ensure fairness. And finally, D, it should be required for all athletes, both male and female. As I said, there is no single right answer here, but this is a topic that athletic events are starting to struggle with more and more as our awareness about some of these issues becomes more pronounced. And then finally, what about Santi? Okay, remember this all started as a case study of Santhi. We kind of went off, talked about meiosis, gender identity, and stuff like that. Well, Santhi was actually stripped of her silver medal. The report was never officially released. The medical issues are private. However, an anonymous official is known has reported that Sanji's test revealed more XY chromosomes than are usually present in a genetic makeup of a female. No official statement has been made. That suggests it's very likely that Santhi was XY. But as we have seen, it is entirely possible that Santhi is XY, SRY minus, in which case developed fully female, was not trying to con any judges or anything, did not have a biological advantage of higher testosterone levels or something like that, and yet was still stripped of the silver medal. Santhi actually, just continuing the story for a minute, even though it's not written here, Santhi was banned from the sport for quite a while, was not able to compete at all, was not even able to get a job, actually ended up working in a brick making factory, living at the poverty level. However, after some time, Santhi got a job as a coach and currently coaches other runners and stuff, helping them and training them. So my hope is you enjoyed this way to introduce some of these concepts and look at how they apply to real world situations. The answer of are they male or female can be much more complex than you think it would be, and understanding the biology underneath can help you understand how complex some of these topics are. Okay, hope you enjoyed it. If you have questions, make sure to bring them to class.